Introduction to Microbiology. This lecture mainly concentrates on Introduction to Bacteriology. So what's microbiology? Microbiology is study of microorganisms. So we have two types of pathogenic structures in nature. One is in cellular form, another one is particle form. The cellular organisms which causes infection in humans are bacteria, parasites and fungi. The acellular forms, which are most of the time we'll call it as particles, they are virus and prions. These microorganisms are very tiny in nature. That means bacteria mainly. The lecture concentrates on bacteria. So the bacteria are very small in nature. They are 1 micrometer in size, very 1 micrometer in size, which is not visible with our normal eye. We can observe bacteria with light microscope or either with electron microscope. It is not visible with normal eye. Very small, tiny organisms. And the bacteria are considered as similar size as mitochondria. The first type of cells on earth are prokaryotes. The life on earth started 4 million years ago according to the records and fossil records. The recently found fossil in Australia, near to Australia and Greenland, suggests us the prokaryotic type of cells were there since 3.5 million years ago. So up to 2 million years since then, the only life on earth are prokaryotes. To classify this life on earth, cells on earth, in 1980, Woos defined the 3 cellular domains. Those are bacteria, archaea and eukaryotes. 3 domains: archaea, bacteria and eukarya are eukaryotes. Archaea and bacteria are prokaryotic type of cells, but they're different in nature. Their RNA sequencing, their cellular membrane structure, cell wall structure is entirely different. Whereas eukaryotes, they have very well defined nucleus and they will have membrane bounded organelles. In this eukaryotes, we have protists, fungi, plants, animals. So the best example for protists are algae and protozoans. Whereas fungi, there are different varieties of fungi which are useful to humans and some causes infection. The best known useful fungi is mushrooms and plants and animals in eukaryotes. So archaea are prokaryotic type of cells, but they are different from bacteria. So they live mostly in harsh environments. That means in hot spring, high temperature areas or high acidic areas like in acid mines, and they can also live in normal environments. In humans also we can see these archaeal organisms, but there is no known pathogens or parasites in archaea. Until now, there is no known pathogen or parasite in archaea. The best known archaea organism in humans is M. smithi, Methanobravibacter smithi in gut. It is normally helpful to humans in digesting of food. Archaea are useful in producing biogas as well as creating sewage. So bacteria are prokaryotes. Prokaryotes means no nucleus, that means no nuclear membrane. The nucleic acids are spread all around the cytoplasm and they won't have any organelle at all, any organelles. And the cell membrane lacks sterols The cell membrane almost similar to the cell membrane of eukaryotes, but they lack sterols. In humans, the cell membrane consists of cholesterol, but the bacteria lacks any kind of sterols in cell membrane. And they have a circular DNA. And the ribosomes have 70S in nature. Normally in human cells, we have 80S ribosomes. And the size of the bacteria, the biggest size of bacteria is 1 micron. The biggest bacteria is 1 micron. And we always have pathogenic organisms smaller than 1 micron. So the difference between the bacteria and Archaea or Archaebacteria. So the bacteria, we can have a peptidoglycan layer that is cell wall, that is murine in nature, that means very rigid, murine in nature, and which is made with muramic acid. They can have 60S RNA, and this RNA sequencing, depending upon this RNA sequencing, the Archaebacteria and bacteria are entirely different. Whereas Archaebacteria, you have a pseudomurine in nature and no muramic acid in peptidoglycan. So the major difference between these three domains, eukaryotes, archaea and bacteria. In eukaryotes, you'll have a nuclear membrane, whereas bacteria and archaea, they won't have eukaryotic nuclear membrane because they are both are prokaryotes. And the chromosome shape is linear in nature, whereas bacteria and archaea, they are circular in nature. Whereas cell membrane sterols you can have in eukaryotes, but they are absent in both bacteria and archaea. The ribosome size is 80S, whereas the bacteria and archaea, they are 17 size, but they are different. The RNA sequence is different. Organelles, you can have a lot of organelles in eukaryotes, whereas the organelles are absent in both this bacteria and archaea. And the amino acid initiating protein synthesis is through methionine, whereas in bacteria N-formyl methionine, whereas in archaea is same like eukaryotes. So taxonomically, how we can organize the bacteria, how we can classify each and every bacteria. So in 2001, there was one scientist called Burgee, and he first started a manual of systemic bacteriology. We'll call it as Burgee's Manual of Systemic Bacteriology. According to this, we can classify each and every bacteria. So in this, for example, we can take E. coli, Escherichia coli, which normally seen in human gut, which is the most common cause of urinary tract infections in females. So how we can classify this E. coli or E. coli? So the domain of this E. coli is bacteria. It comes under bacterial domain. The phylum Proteobacteria, the class Gamma Proteobacteria, order Enterobacteriales, family Enterobacteriaceae, genus Escherichia, species Escherichia coli or E. coli. Coming to the history of microbiology. In 1665, Robert Hooke is the first man to observe the cells. He observed the living plant tissues with 2x magnification. So when he observed these plant cells like cork, he observed little boxes which afterwards referred as cells. He used very simple magnifying glasses, and he suggested that all living things are made of cells. And this is the microscope that was first used by Robert Hooke. And with his observations, with his book Micrographia, which is written by Robert Hooke, the other person who got inspired on this, observing the small tiny things is Anthony von Leeuwenhoek, who is a Dutch scientist. He works in glass factory, and after reading this book he was inspired and he started to observe a tiny things with magnifying glasses. And this is the first microscope which was developed by Anthony von Leeuwenhoek, and he observed bacteria, and which he was used to call it as animalcules. And he observed protozoans, sperm cells, blood cells, and microscopic worms. Later on, the big man who was called as father of microbiology is Louis Pasteur. He started to propose one theory called germ theory. That means, according to this theory, any infection, any disease is caused by germs. Germ theory. He started to propose the germ theory. Later on, it was proven by Robert Koch, who was his associate, and he explained about fermentation. He explained about pasteurization of milk, wine, and he developed a rabies vaccine. Robert Koch, who was a colleague of Louis Pasteur, he was a doctor, and he explained the postulates of germ theory. In the next PowerPoint next slide we can see postulates. He identified the microbes anthrax, tubercular organisms, cholera, and he developed the microbiological media where we can grow the bacteria outside of the host, and he developed pure cultures with stick plates. So Koch postulates, what he explained, what is the germ theory? Depending upon this Koch postulate, the germ theory was proven. Specific microorganism is present in all cases of disease. So according to this, any lesion must have the microorganism which causes that lesion, and organism can be obtained in pure culture outside of this host. So the organisms which causes that lesion has to be extracted, and it must grow in cultures outside of host. And after growing, the organism, when re-inoculated into experimental animal, it must cause the same symptoms, same lesions. The organism can be isolated in pure culture from experimental infected host. So whatever the infection that occurred in that experimental animal, from that same lesion the organism has to be extracted, and it must be grown in pure cultures outside of that experimental animal. So depending upon these four postulates, 4 points, he proved the germ theory. So bacteria. So whenever we have a bacteria to grow on culture media or inside the humans or inside the host, it must have, whenever we say bacteria, it must have few important things. One is nucleic acids, we can call it as nucleoid, ribosomes, cell membrane, cell wall, and with some other layer on cell wall. So the structure of bacteria. Structure of bacteria. So the bacteria will have a cell membrane, a cell wall, a capsule or slime layer. They will have flagella. They will have pili or fimbriae. They will have mesosomes. They will have ribosomes, inclusion bodies, plasmid, and DNA or nucleoid material. So this was a diagrammatic representation of a bacterial cell, which consists of a capsule, a cell wall, cell membrane. Inside it will have a inclusions. They will have a small